I'm really happy to be able to talk about this. I feel that I'm a newcomer to yeast, but I became very interested in yeast that might be associated with insects that lived associated with fungi. And I have to thank Clete Kurtzman, who very early gave us a huge database of all the yeasts that were sequenced so that we could be able to work with this group that was pretty foreign to all of us, except for Sue. Now, Sungwei Sue came to the lab supposedly for 6 months to learn to do some work in the field, and he stayed 12 years and is still in the United States at ATCC. Hector Abina is shown in the middle, reading a Thai newspaper. I thought he was going to be here. He was here earlier in the week, but later you'll see his face. And then Jennifer Langasard, who's been very, very helpful to us for collecting in Thailand, is shown there with, of all things, a beetle costume for a dog. Now, we began to be interested in this because a number of years ago, Dr. Prillinger, who was here earlier in the week, had published some papers, and he showed that there were basidiomycete yeasts that were growing in the hymenium of certain mushrooms. And so I'd always wondered if perhaps they might not have got there by being carried by insects. And so we began to look at this with the database of yeast. And also, we were already focusing on some of these fungi and insects because we were collecting lobobiniomycetes when Alex Weir was in the lab. And so we could have a diverse group of beetles all going to mushrooms so that we could have targeted resampling. And then the main reason for starting this project, as I alluded to, is that Sungwei Sue came to Baton Rouge. Now, Sue was very good at developing a number of techniques. And so by looking at many studies that had been done, we decided we wanted to look at the inside of the beetle. And so he had a protocol by which he was able to disinfect the outside of the beetle and then collect the material that came off of it so that we could see what was on the outside of the beetle that survived and maybe what was on the inside of the beetle that was different. So this worked very well. A friend of mine calls it coleoscopy because we were looking at the beetle gut. Now, about after we had started working on this project, Hunt and his colleagues came out with a very large phylogeny of beetles. And I don't know how well you can see, but up at the top part of the tree, which is a continuation of the bottom part of the tree there are a lot of hot pink areas. And this indicates that fungal feeding occurs in these groups of beetles. And I've outlined some of the ones that are particularly important: Nitidulidae, the Erytilids, Endomycids, Ceads, Tenebrionids, and some of the others that are in the more earlier derived lineages of beetles. Now, I should point out that the ones that are the more recently derived ones probably have more specificity with the yeast because of their morphology, their gut morphology, and I'll talk more about that in a minute. And so we have about 30 families of beetles, and here is a variety of the beetles showing some of the ones that we worked with. They're very, very pretty. So these, there's 7 families shown here, but there are, as I said, many more. One of the important families is Tenebrionids, and of all the Tenebrionids, there is one subfamily, the Diaporini, which are associated almost primarily with mushrooms their entire life. And you can see these. They're often called the false ladybird beetles, and you can see why. There are also a number of other beetles, also brightly colored, really pretty beetles, that are in the family Erytilidae. And there are over 5,000 of these worldwide, it's estimated, that are described. Now, if you look at the yeast gut, either you have Sika or not. And the Sika that we were looking at occurred here in some of these more recently derived beetle lineages. Here you see an anabiid and an Erytilid. And these are outpockets of the anterior end of the midgut of these yeast. And we find that many times these cica are populated by almost a single species of yeast. Also, we looked at a few other beetles that were not always associated with mushrooms or fungi, but were associated with wood. And these basalid beetles seem to have the yeast throughout the gut, but mostly concentrated in the posterior hind part, which is what had been found by Lickford and his colleagues who were looking for trichomycetes. Now, in the first year that we were doing this study, Sue put together some amazing slides. Here is our D1, D2 genotypes. This is the large subunit ribosomal RNA gene. And you see that here we've got yeast that were known to be identical from the D1, D2 to other yeast. And then we had some others that were up to 5 base pairs different. And then we had the majority of our yeast shown in red here. And these were more than 5 base pairs different from known species of yeast. And usually, I should say, these were not just 5 base pairs or more different. They were much more. Sometimes 30 and 40 was not unusual. So they were very distinctive. And if you look at this tree, you may be able to see the red dots. And they occur in kind of clusters. And these are the things that we were isolating from beetles. And there are a few marker species there in amongst the clades that we were sampling. And we mostly found 4 clades of yeast that I'll mention today. We found others, but they're not associated with beetles. They're associated with other kinds of insects, or they were not so, so large numbers as the ones that I'll mention. Now, our largest group was about 30% of all the yeast that we isolated were in the Candida tanzoensis clade. This was described from a single species of a single isolate, single strain of yeast that was isolated in 1966. And Dr. Nikasi, after waiting 22 years, decided to go ahead and describe it as new at the time he was retiring. And then about the time we started isolating all these tanzoensis clade members, Cleet Gertzman, he described 6 species. And then we found a large number more, and we described 16 of these. And so having this one marker there was very important for us to realize that we had something kind of good. Now, here is a slide that shows the beetle associations of the tanzoensis clade members that we were able to collect, and some of the ones that had been collected before. The ones that came from beetles that we were able to target and retarget are in the maroon and the lavender colors, and these are species of beetles that were in the Tenebrionidae and Erytilidae. And here you can see some of these Tenebrionidae beetles that we got yeast from, and you can see that there's some degree of specificity, although it's not very strict, and it's less strict now that we've collected more. But there's some, such as Boletotheris beetle, that has the Candida boletotheri, and this was known through a large portion of the United States, the eastern part of the United States, where these beetles live in Ganodermas. So there is some specificity. We found that tanzoensis occurs in North America. It occurs in Central America, where we collected in Panama, and it's also been collected with Jennifer in Thailand. And you can see some of the species are in the same place, but there are also others. But I think that probably with more collecting, this will go away. Now, the Candida cruisii clade seem to be associated with nididulid beetles. And Andre Lachance has worked with nididulid beetles that have a different behavior. They're associated with ephemeral flowers, and they seem to have very different yeast. But here in these cruisii clade members, we see that the blue arrows are North America and the reds are Central America, Panama, one small island in the middle of the Panama Canal. And we even get the same genus, Pelotes, that occurs in both the United States, the southeastern United States, and Panama. And you can find that some of these same clade members are there. So again, we have some level of specificity. And here we have another clade, the Myrozyma gurielmii clade. And these members were associated with a number of different beetles. Some of the ones that have the Zika, but others that don't. But most of them are beetles with Zika, and they're in this more derived group of beetles, the later diverged ones. And then we also have Streptomyces stipidus. That was one of the things that we found lots and lots and lots of times. We've done a lot more with these beetles than I'll indicate here, but just to tell you that we have some of these beetles that have been looked at for bacteria with some people at the Lawrence Berkeley lab. And this was all because of, these are species here, of Streptomyces that we've collected and some that were described by other people. And here is another clade, Bathospora, which also comes from some of these wood-eating beetles, including Psallidae, but others. And this is New Nguyen, who was a student, an undergraduate student in my lab, who worked harder than any of the graduate students. And he published a number of papers, and he was the one that discovered Bathospora and worked at it to make sure that he got ascospore formation. And New has gone on and recently got a PhD with Tom Bruns. So we've got two species probably that go in this clade that are still as candidates here. And I'm going to go back a minute because I want to point out that this Streptomyces has been restricted, it's more restricted by Sue and some of his colleagues. Hector and I may have been a bit exuberant, and Sue and his colleagues used more markers and also a more inclusive taxon sampling. And so they find that what we thought was one Streptomyces clade actually turned out to be four, that some of which they've described. And there still remains to be a reanalysis to see. One of the shortfalls of what we've done, most of the markers are the ribosomal genes, although Hector has used for the Streptomyces and Bathosporas, he's used some more markers as well. And I just want to show here the yeast clades associated with phthalates that Hector has compared in the yellow from the United States and the blue Thailand. And so it looks like there's a lot of diversity that he's discovered in more tropical regions that has yet to be worked on. And some of the members of the clade are xylose fermenting, which was something of his interest, but there are also some that don't. And there's a summary, 300 novel genotypes from these yeast, from these beetles. And I just want to acknowledge at LSU we had many, many undergraduates, especially New, and 3 postdocs who have worked on the project, Sue, Hector, and Kevin Jones that I didn't mention. We've also been lucky to have funding from the National Science Foundation for about 8 years for this project. Thank you.